Here we are again in your subject microbiology. And last time we were talking about the different highlights or views on microbiology. Today we're going to discuss the history of microbiology, the persons involved in the evolution of the study of microbiology, and also likewise, let us try to highlight some of the contributions of these personalities in microbiology. The history of microorganisms could be traced as early as when the cells were being studied. Since microorganisms are considered as one cell, then we could consider that the discovery of the different concepts involving the cell theory, that is, all living things are made up of cells, could also imply that it is a recognition that microorganism as a unicellular organism is a living organism. So, with the concept of cell theory, we have to thank Sir Robert Hooke for sharing to us his knowledge in cell theory. So, Robert Hooke had first discovered the little boxes and he considered this as the cell. Later on, more rigid study using more appropriate lenses and tools in microbiology had allowed Anton van Leeuwenhoek to see the living organisms. And using a 300 times single lens microscope, quote unquote, he had introduced to us the shapes of different microorganisms. So if you're going to look at this is the illustration of Anton van Leeuwenhoek on his observation of microorganisms as little boxes. So he had identified figure A as this, and then in figure B, you could see the illustration that these organisms are actually moving, or these little boxes that first Robert Hooke had identified could move from one point to another. And then these are the shapes of the little boxes that first Robert Hooke had discovered. So here are the circular, here is more of a linear, and this one is already with curves. And then he had identified these small cells as we animalcule, and we consider this nowadays also as the microorganism. This was how the microscope that Leeuwenhoek had used in his magnification or in his study of microorganisms. It is not the microscope that you are familiar with today. As you can see, it is just a tiny lens in which it could be used as this. But if we're going to look at the parts, it speaks of the salient parts of a microscope. So here are the lenses. So the lenses could mean your objectives as well as your ocular piece. And then the specimen holder refers to our slide. And then the focus screw could mean our coarse adjustment and the fine adjustment. And then we have also the handle. So there is the function of the handle. But today, there are some microscopes that do not have also handles. Like for the case of the electron microscope, another microscope that entails high magnification and that the sensitivity of the lenses will not allow or advise for movement from one place to another. Okay, let us try to go back to some great names in microbiology. There are people who had worked on the spontaneous generation, but there are also some people who had worked on to disprove the theory of spontaneous generation. So, Francisco Redi, Sir Rodolphe Virchow, Louis Pasteur had developed the theory of biogenesis, claiming that cells can only arise from pre-existing cells. So, you try to look at the date in between discovering really if spontaneous generation is true or not. From 1668 to 1860s, the theory of biogenesis was developed. And then germ theory of diseases was also contributed by Louis Pasteur and Robert Koch in the study of microbiology. They disproved some social notions that organisms are the cause of many diseases and not by sins, bad character, poverty, or a sort of a punishment, but rather from microorganisms. Louis Pasteur had also showed and explained to us the process of fermentation, that is in the production of some foods and other basic needs that we need, and also likewise illustrated how spoilage had occurred due to the presence of microorganisms. Of course, as it was made mention, he was also one of the many great scientists that had disproved the spontaneous generation of microorganisms. And pasteurization, from the word pasture, he had discovered that one way of killing microorganisms is using high heat at high pressure at low duration or short duration. Luckily, Sir Louis Pasteur had given us the germ theory of diseases, and this we will discuss later on. And then Sir Louis Pasteur was also the one who had developed a rabies vaccine. Now let us try to look into the spontaneous generation and how scientists had looked into this. Early belief that some forms of life, living organism, could arise from vital forces present in non-living or decomposing matter, meaning any form of thing could generate life. That was the theory of spontaneous generation, saying also that life is spontaneously developed from non-living organism, but abiogenesis have told us that living organism could only be developed from living organism, or a specific cell is generated from a specific cell. So that notion or that theory had generated more ideas about microorganism. Now, one great work of Sir Louis Pasteur that had showed that non-living organism could not generate living organism is his study or experiment on the swan neck blast. This is the real swan neck blast or the ghost nest blast that Louis Pasteur had used in his study to disprove theory of spontaneous generation. Through this blast, he had placed some boiled meat infusion, and then this meat infusion is a non-living organism. Then, in this portion, air could flow, hence air could carry the microorganism. But there are some vents present in this ghost neck that will not allow the entry of foreign organism or foreign substances that could probably carry microorganism. So what happened? These microorganisms were just simply trapped here, not allowing to go into the non-living boiled meat infusion. Of course, expected, the next thing to happen is that the flask were not able to generate microorganisms, as shown in this illustration. But another experiment have been done, that is to cut this ghost neck area of the flask. Since this was directly exposed to the environment or through the air, microorganism was able to develop. But that does not mean that this had generated microorganism. because when this broth was still covered, there were no microbial growth. But when these flasks have cut and exposed to the environment, microorganism had developed. And then we have the germ theory of diseases, or the germ theory of disease. Still, Sir Louis Pasteur had developed this together with other great scientists, and took a better understanding of the germ theory and cell theory's relationship to displace the spontaneous generation. So what happened is that they had seen that we animalcules could possibly present in any space. However, there were some instances that this should be developed through the presence also of we animalcules. That it is not caused by demons, it is not caused by witchcraft, it is not introduced by bad luck, neither as a punishment by God, but rather it should have been coming from the same organism or same living organism that was found to cause a disease. This could be found in another concepts in germ theory that is contributed by Koch postulate. We will learn more about Koch postulate later on. So from here, we could also consider that there are some things that is not true to the Koch postulate, no matter how true is this. Some organisms have never been grown in the pure culture on artificial media. That is one. If you could remember, one of the statement in Koch postulate is organisms should be grown or could be grown in pure culture media. But science of today will also say that this is so limited. The Treponema pallidum was the first microorganism that scientists tried to grow in pure culture in order to study its treatment. And luckily, there are some scientists that were able to discover the treatment of uh, Treponema syphilis. Another microorganism that brought scientists into interest of growth on pure culture is the Mycobacterium leprey. It is responsible for the disease leprosy. The scientist that had discovered the Mycobacterium leprey is Dr. Hansen. And unluckily, Dr. Hansen is a scientist that had met its fateful day because of the action of leprosy. Dr. Hansen himself, because of exposure to microorganism Mycobacterium leprey, became a victim of leprosy. So it's just like Madame Curie. He discovered, yes, you are right to that, and also died of this uh, metals. And then you have Joseph Lister. Sir Joseph Lister had worked on medical settings and had really worked on the importance of disinfection of hands and also the use of heat for sterilization. That is to prevent infection in many medical allied activities. And of course, this is Sir Robert Koch, the first to prove that bacteria actually diseases or microorganisms are responsible to most diseases. That's why he had given to us the Robert Koch postulate. So he was the one who introduced to us the etiology or cause of various infectious diseases, the pathology of infectious diseases. Robert Koch had also worked on anthrax, tuberculosis, and also cholera, and also worked on the pure culture on tuberculosis in which earlier scientists could not do it. So he also, that is the reason why he claimed that microorganism could be isolated as pure culture because of his famous work on tuberculosis. And also likewise in a microorganism found in animals, the anthrax. What are the exceptions to Koch postulate? That is, it was claimed that uh, he was the person who worked on to use other forms of animals to understand the action of microorganism. So he had introduced microorganism to animals in order to look into its probable action to human being. This is actually our basis for ethical researches in animals and human. Koch established, while Koch has established the etiology of microbial diseases, these are the three important diseases that we are still working on until today. The cholera, which is a fecal to oral diseases that is usually caused by microorganism Vibrio cholera, and the common symptoms is of course the presence of diarrhea. And then you have tuberculosis. The microorganism responsible to this is Mycobacterium tuberculi. So it is more of a respiratory infection, just like your COVID vaccine. And then, COVID vaccine, COVID disease. And then you have the anthrax for sheep and cattle. The Bacillus Anthracis is the microorganism responsible to this. So these are all bacteria in which their characteristics are still used today to study the reactions of newly discovered microorganisms. Let us try to look into Bacillus anthracis. The Bacillus anthracis case or study in microbiology had become a great reference in agriculture, especially for the case of diseases in animals. To, to this day, we are actually suffering from uh, pork shortage or meat from pig shortage. And the swine flu, though it is viral, um, the history of Bacillus anthracis was used to look into how could this be prevented or mitigated. But what is this Bacillus anthracis? Bacillus anthracis is a microorganism that belongs to gram-positive bacteria. Of course, they are non-motile, so you could easily destroy because you could easily attack. And then it is aerobic, so it is exposed. It is a spore-forming rad, so it is easy to identify. The Streptobacillus with central spores also is one possible cause of anthrax. And here, there is an additional knowledge for the presence of spore. So we are trying to make the microorganism or the disease more complex by adding more characters that we should prevent or study in order to get away from this disease. Because if you do not know the structure of the microorganism, if you do not know the nature, the cause of that microorganism, its etiology, its morphology, that microorganism will never be eradicated. That is how beautiful is medication nowadays. You should know what you are boxing with in order to kill that microorganism. So we know very much that anthrax is a disease caused by livestock, and we fear so much that humans could be possible victims, but no. The reason why we are so particular with this is humans are active carriers of possible uh, things that carries this anthrax, because we are exposed to what are usually exposed to these microorganisms. We are carriers. So this is how the anthrax looks like under the microscope. That is a stained microorganism. There are three forms of human anthrax, by the way. One is external, that is the cutaneous anthrax. It enters through cuts or just simple abrasion. And of course, the ulceration is necrotic, and it is painless. However, the mortality rate is 20% if it remains untreated. Another is through the gastrointestinal system, also called gastrointestinal anthrax. So we take in this through contaminated meat, and these are the common symptoms: abdominal pain, fever, vomiting, especially with blood, and then severe diarrhea. Mortality rate is 25 to 60%. And then we have also inside an anthrax attack through inhalation. This is more a potent type of anthrax. So initial symptoms resemble to just simply common colds, and then there is severe breathing problems, and then shock, and then death if it remains unresolved within 1 to 2 days after the onset of acute symptoms. And the mortality rate is 99% if it remained untreated. Well, that's so much. Then I would like to show to you some claims on anthrax as biological weapon, and I would like you to make some reaction to this as your assignment. Please take note that we have made mention that anthrax is deadly if not treated early. So that is what we would like to happen if we go to war. That is to cause death, and that uh, early symptoms should not be treated in order to cause death. Spores can be produced in large quantities. So we want that these weapons that we have should be developed in large quantities, not limited. And then another is they are viable. They remain to be strong for at least 60 years. So let us see your lifespan. Wala pa kayong 60 years. But the spore of an anthrax, if developed as a biological weapon, could be with us for 60 years. And the symptoms related to this disease will be with us. Spores can be spread, of course, that's what we like. And this could be carried through missiles, rockets, bombs, mail, and even just simply crop dusters. And of course, this is another thing that we like, or biological weapon producers like. It should have no odor, no color, no odor, no taste, and most of all, it should not be treated early. It, since we could not see, we could not smell, we could not touch, then giving treatment to it might be delayed. So it made mention here that if if antibiotics is not effective, it is not administered early within 24 hours. So, my question to your assignment or case analysis is, if you will be given a chance, and it will promote you to become a general, will you divulge your findings or the result of your study that you had developed anthrax that is good for biological weapon? So imagine now that you are a scientist, you are a microbiologist, and through your work, you were able to develop a microorganism that is similar to anthrax that could be used as a biological weapon. Will you make it as a secret, or will you gonna sell it to develop this as a biological weapon? Okay. That is your first assignment. You could get as many as references that you could get online if you wish. Okay, that is all for today. Thank you for bearing with us again with our limited knowledge or limited way of sharing you our knowledge.